Welcome, everybody, to the January 25th meeting of the IMAG-MSM Working Group on Multiscale Modeling in Viral Pandemics. I appreciate people coming for the seminar today. If you need to unmute and you're not able to unmute for some reason, put it in the chat. Sometimes the security settings are to prevent Zoom bombing occasionally. That makes things difficult for people. Today, we're going to have a single talk on B-cell receptors. And as always, I need to remind you that the seminar is live streamed on YouTube and available in lightly edited form for later viewing. As always, Tom and I are available to take your comments, criticism, suggestions, as are Jim Sluka and Lorenzo. Please let us know what works, what doesn't work, suggestions, comments. We have our various communication channels. We could use help getting the word out about things. We have a Slack channel, X channel, the main IMAG MSM wiki page, the LinkedIn page, which seems to get a lot of views, even if it doesn't lead to so many people coming to the seminars. And of course, our growing archive of YouTube videos of more than 150 now. We have the GLIMPRINT page. Tomorrow we're going to be talking about that a bit. I hope people can help us develop that as a resource. We have that MSM viral handle as a tag. Please remember, if you're doing things related to the multiscale modeling, to tag with that. Help us get the word out about this community. We have a couple of communities. Lucas, I think did you want to talk about this announcement?
Maybe very briefly, yeah. So this is an online workshop that we will hold on February 19th. And so all of you who are interested in forecasting heuristics and infectious disease surveillance are invited to join. And I think it might be also of interest to some of you who are thinking about applying for one of these interdisciplinary grants between applied math, infectious disease modeling, and behavioral sciences. So yeah, please feel free to join or reach out to me if you have additional questions.
Great, thank you. It's exciting to see that coming up. I have two announcements on my own behalf. The first one is that Indiana University is going to be conducting a multi-year search for a significant number of positions in broadly defined biomedical engineering, bioengineering, computational biology, computational bioengineering, digital twins are all areas of interest to us. The formal announcement will be coming out in the next day or two. We're a little bit late this semester to be starting, but these could be junior or senior hires, potentially cluster hires as well. We're looking, I think, to hire 3 people this year. And so if you're interested in that and you want more information or you want to pass that on to people, please do reach out to me. I'm the chair of the search committee. The other thing I wanted to say is that I'm involved in an NSF project to build communities for multicellular, multiscale modeling, what I'd call virtual tissue modeling. At the moment, we're trying to do discovery of the community. And so we are doing short half-hour interviews with people who develop multicellular models, either use them, apply them, regulate them, fund them, or the software packages that enable them. And so if you would like to talk to me about that or be willing to talk to our team more generally, again, please mail me, and I'd be very grateful if you do that. If you have ideas about important people that we should talk to build this community, I'd also be grateful for suggestions. Could be quite broad thinking. It could be somebody in EPA, somebody in a pharma company. Doesn't have to be somebody who's necessarily doing multi-cell modeling directly at today, people who might benefit from it in the future as well. NSF has a sort of a European style glue grant called POSE program to build open source communities. And this is really about trying to do that for this multicellular virtual tissue world. Are there other announcements from the audience? We have a steering committee meeting tomorrow at 11. If you think you should have been invited and you weren't, please let us know and we'll get the invitation to you. We're going to be discussing trying to have a first kickoff meeting for GLIMPRINT. We'd very much appreciate people's help in designing that and getting it going. Any other announcements? No. Okay. Coming up, February 8th, we have AI-based computational modeling to support drug development against autoimmune diseases. February 22nd, we have a speaker from the Netherlands eScience Center who helps teams develop open source and HPC applications. March 7th, we have Belinda Akpa from Oak Ridge, who's trying to do very large-scale multi-scale modeling, basically from molecule to whole organism phenotype. We have an open slot on March 21st, so if people would like to suggest, as always, you've heard me say it before, I'd like to suggest yourself, your students, your postdocs, the people you'd like to hear from. Please let us know, and Lorenzo will do his best to try to get them on the schedule. If you spoke a year ago and have new work that you'd like to present yourself, you're always free to come back and speak again or have your students present. So please do take the opportunity to give us some suggestions for speakers. We'd be very grateful for that. Anybody you'd like to hear. And I don't want to take more time from our speaker today. We're very pleased to be talking about a real immune problem in a modeling context. And so I will turn the screen share over to you.
So hey, so thank you for inviting me, and thank you for coming. So I'm going to talk about the work that we did within the University of New Mexico STMC. STMC stands for Spatial Temporal Modeling Center. Was basically a long 10-year P grant. It was a center that I mean, the people are still there. It looks at not only systems biology type modeling with an eye to actual spatial aspects. Right? And so what I'm talking about today is a spatial stochastic model of the pre-B cell receptor. This was, so I'll finish with acknowledgements, I hope, okay. So this is just for myself. I hope I will go through all of these. So this has to do with PB cells, have to do with leukemia. I apologize if you will find the next few slides as overly simplistic. It's meant to be that I'm myself a physicist by training, so I will just go through these just very, very superficial headlines. So leukemia in general is a proliferation of dysfunctional white blood cells. And it impacts immune function, which is essentially done by the white blood cells, but it also ends up undermining the oxygen transport function of the blood by crowding out the red blood cells. There's various types of leukemia. The two big divisions are lymphoblastic, and I don't know what ML is for, but the two major types, ALL and CLL, acute and chronic lymphoblastic leukemia, involve malignancies based on lymphocytes. And what happens is that lymphocytes, in the process of their development, they basically stall and do not progress through mature stage, but also do not go through apoptosis. So they just end up accumulating. And in this talk, we are concerned with one specific type of so-called precursor B acute lymphoblastic leukemia, or precursor B ALL, where the immature cells that accumulate are immature precursors of the B cells. This particular type is the most common form of ALL, and it accounts for a very large fraction of newly diagnosed pediatric cases. So it is of kind of immediate and acute importance, as always. So I have to go into some details about the B cell signaling. This is a model for pre-B cell receptor signaling, but of course that is kind of related, and the major elements are the same as those for mature regular B cell signaling. So B lymphocytes are central to the immune response machinery. Mature B cells have receptors, so-called B cell receptors, or BCR, which are kind of outlined in this graphic that I got from Wikipedia. And the mature receptors will have antigen-specific binding regions outlined up here. The other feature, yes. Other features of the B cell receptor is that the main body, as it were, consists of something called the heavy chain and the light chain, and then the light chain is the one that represents or contains the antigen binding function. In addition to that, I think that's on the next slide, there's also this signal transduction region. So what happens is that the antigen binding site is obviously the one that interacts with antigen, and it is actually pretty specific. And in the presence of antigen, these multiple BCRs will bind to the same antigen and form these aggregates. And that's the first step in the signaling of mediated by B cell receptors. Now, the signal initiation itself is initiated by a pattern of cross-activation that requires not only the presence of multiple BCRs bound together, but also the presence of other kinases which will bind to the BCRs and then follow a pattern of cross-activation, which I will go into more details. Importantly, there's two types of these helpers. They're both kinases. There are several types of so-called Src family kinases, or SFKs. The best known from among them is Lin, and that's the one we include in this model. And another kinase, which is the one that drives the downstream signaling, and it's kind of the endpoint of our approach, our model of this, is Syk kinase, which, activation of Syk is sort of a measure of the downstream signal. So as I mentioned, if you look at this diagram of the B cell receptor, so there is the antigen binding site, which is essentially sitting on the light chain. The kind of the receptor part, as it were, consists of the light chain and the heavy chain. And in addition to that, there is something called a signal transduction region, which consists of two additional, I think these are the receptor tyrosine kinases. So these two kinases form a dimer, and it's kind of bound, it's part of the receptor, right? And so when it functions, I'm missing the figure here. So when it functions, basically the presence of antigen causes the formation of these aggregates of receptors, and then the multiple aggregated receptors together with the kinases will go through this activation process, and ultimately the activation involves these two, the IgA alpha, IgA beta dimer. Okay, so now how does this relate to pre-B cells? Because we're talking about precursor B cells. Well, precursor B cells are basically B cells that are, 2 steps before the emergence of the version of the B cells that has the mature, the regular B cell receptor. The major difference is that receptors on pre-B cells or pre-B cell receptors do not have the proper antigen binding region. Instead, the light chain is still replaced by something called the surrogate light chain. And this renders the receptor dysfunctional or unfunctional, essentially, as in terms of binding antigen or anything else. So the difference between the pre-BCR, the pre-B cell receptor, and the regular, the mature B cell receptor is that the pre-B cell receptor is a B cell receptor which has the signaling part, but it doesn't have the antigen binding part. As a result, the main thing is that we'll go into details, but the main feature of this is that the pre-B cell receptors are unable to form these big chains, which result in the signal that is part of the job of the B cell. Right. However, during the so this is what I so however, even though this pre-B cell receptor version of the receptor is unable to bind antigen and make these stable chains, there is a low-level signal that is actually expected to be present also in pre-B cells. And the presence of the signal is actually a requirement for the cell to be allowed to develop further, right? So this low-level signal exhibited by pre-B cells is called the tonic signal, and it's actually required for the survival of these cells and their progression to mature B cell stage. Now, the problem is that some B cells, the malignant B cells, the ones that end up accumulating, have this feature that somehow they survive this checkpoint. So somehow they emit the tonic signal, but something else is wrong with them. So they are actually unable to proceed to mature stage. So they just accumulate and cause all the problems and the phenotype of leukemia. So the point of this talk is a model that is meant to investigate the mechanism of tonic signaling by these malignant or by some malignant pre-B cell lines. Now, this is actually a continuation, or it's linked to a previous paper by the same group. So as I said, this is the SDMC has two, or at the time when this was started, had two experimental groups and a theory part. So one of the experimental groups had worked on this on a number of leukemia cell lines collected from patients, and they used primarily this single particle tracking to look at the movement and dynamics of receptors on these cells. So I'm sure everybody here knows, but just in case. So you can take cells, but usually abnormal cells. You can take cells and grow them. And so you can take a cell line and basically have it there and investigate it and look at it under a microscope. So this single particle tracking, there's no additional explanation of it, but consists of actually tagging, so using little molecular size tags, which are chemically fused specifically to receptors, and then image them. The resolution of these images goes into the tens of nanometers. So you can actually derive trajectories of individual receptors. So you can actually image them moving around. And based on the analysis of these trajectories, you can learn things like, do they get stuck somewhere? So do they get stuck in places? Do they move uniformly? Do they tend to form oligomers? Now, when there's a little technical issue with looking at oligomers, which is that you cannot, so what you can see on the microscope is basically light emitted by these tags that you put on the receptors. However, because of the physics of how this works, you cannot really distinguish two tags that are too close together. So if you have labeled, so a dimer that's made of two receptors that are both tagged is essentially going to be indistinguishable or not reliably distinguishable from a single receptor that's being tagged. However, you can create tags with different wavelengths. So in that case, you can actually see two light sources of different colors coming from, well, either the same point or very near to each other. By the way, oh, I didn't. So long story short, the experimental data that we're trying to leverage here consists of single particle trajectories from pre-B cell lines, from primarily two malignant pre-B cell lines, where the receptors have been tagged with two colors. So you can have individual trajectories, and you can also see pairs of trajectories labeled with different colors, which move around together, indicating that these two things are in a dimer. Right. What you cannot see, you cannot tell whether they're a trimer or a tetramer, because again, you only have two colors. So what the data set has is trajectories and observed dimer lifetimes, right? So as I'm mentioning here, so this data exhibits evidence of dimer formation, as I just described, and they have extracted statistical distribution of observed dimer lifetimes. And one striking observation is that there's these two cell lines, one called 697 and one called NAM6, I have no idea where the names come from, exhibit significant differences in terms of this apparent dimer lifetime. One of them is about almost 5 times longer than the other one. So the bonds, the length, the lifetime of the dimer is 5 times, in one of them is 5 times longer than the other. Why is this important? Well, because remember, the entire signaling machinery of BCRs, but as of many other kinds of receptors, relies on this ligand, or in this case, antigen induced dimerization or oligomerization. The whole signaling machinery relies on the fact that you will have receptors sitting next to each other so they can cross-activate. But these receptors cannot bind anything by construction. And yet, that's what's interesting or important in this experimental result, that there's actually direct evidence that, okay, they're not going to sit together for a long time, but they do form transient dimers. And basically, the point of this whole model is really to see if, okay, so now if we just take this piece of information and everything else we know about B cell signaling, and we put them into a model using realistic parameters, can we get this? Does this system want to get to the point where SIG is activated, that is, where the kinase that controls the downstream signal is activated in a measurable fashion? So the rest of it is trying to get that. Another important feature, though, is this transient confinement. So what that means is that as soon as people started looking at single particle labeling of receptors on cell membranes and their trajectories, they noticed that, well, they basically tend to get stuck in little regions. And I can show you trajectories afterwards. I'm not, it's not in one of my slides. Basically, they run around and then they just bounce around for a while and then they run around some more, then they bounce around. This is also observed if you're looking at if you're running software trying to identify dimers. There's a regime. So there's a regime where the two are just uncorrelated. There's a regime where they're strongly correlated. That is, they're essentially basically at some kind of molecular distance. And then there's an intermediate regime when they're not completely correlated, but they are within a small-ish distance. And small-ish means basically tens of nanometers, maybe 100, 200 nanometers across. So this is interpreted as the signature or as the result of the existence of these confining microdomains. So little regions that for whatever reason, for their, maybe it's their special consistency of different lipids or whatever, they tend to trap these receptors. So the receptor goes in with a high observed diffusion coefficient, then it bounces around, so it seems to the diffusion rate seems to be small, and then it's big again. So transient confinement. So these trajectories, just like pretty much many others of receptors on other kinds of cells, exhibit transient confinement. But why? What does this have to do with anything? Well, if you have a thing that's going to concentrate receptors that are supposed to bind that, and binding these, and these receptors binding together leads to signal, then these little domains that keep them together are likely going to enhance dimerization and signal. All right. So then on to the actual result, actual calculation. So our model has, so we tried to kind of include all of the following. So this is a computational model. It's a stochastic, so it's a simulation. And it really combines, it's really based on BCR signaling models from the literature. So we use a lot of their parameters from others with the pre-BCR specific information that we have from the data from Erasmus. Erasmus is Frank Erasmus, the second author here. He was a grad student at the time. His PhD was the experimental part. The first author, Romika, the model was part of her PhD. Okay. All right. So the following elements. So cross-linking of receptors with the possibility of higher dimer formation. So it's kind of, we allow them. It's kind of an open-ended thing. We include lin and SIC, so one each of the two important types of kinases that are intermediates here. But we also allow full diffusion. So it's not a counting model. So we allow actual explicit formation of dimers, trimers, tetramers with lin and SIC bound to it with additional phosphorylation states. And we allow these things to diffuse in 2 dimensions. Actually, receptors and lin diffuse in 2 dimensions, and then SIC is allowed to diffuse actually in 3 dimensions in a thin layer that's adjacent. So SIC is a cytosolvent. And on top of everything, we also add transient confinement. So we have runs with confinement and without confinement. And so the rest is, I'm just going to describe the model, and it depends how much time I have, I think. I'll just go fast. All right. So first of all, the species. So we have 3 base species. Oh, and did I actually mention that? Where is this? Oh, okay. I should have put this little slides here, but this one. So we have the pre-B-cell receptors, but they have to have the following features that need to be put in the model. So the receptors are a single molecular feature, but they have the following, they can do the following. So they can cross-link with other receptors bivalently. So basically each receptor has two receptor binding sites that can bind to other receptor binding site on to receptor binding sites on other receptors. But they cannot form a doubly bound dimer. So they can only form chains, right? Now the items, remember the Ig alpha and Ig beta were basically the signaling subunit of the receptor. So we need to look into some detail on that. So each of these two subunits, they're distinct, they're not identical. One is called alpha and beta. So the alpha subunit, so both subunits have two phosphorylation sites, so 4 phosphorylation sites altogether. We're trying to kind of limit the proliferation of things. So we do not model the sites separately, but the 2 sites per item separately, but each item has 2 phosphorylated states. So each item can be not phosphorylated at all, once or twice phosphorylated. Okay. And then the item sites also serve as binding sites or docking sites for the kinases, for the lin and SIC kinases. And their ability to bind, so the strength of the binding is also dependent on the phosphorylation state of the substrate of the item and of the kinase that binds to it. So now about the kinases. So there's 2 of them. So the lin kinase, so in general, in terms of big picture, as I said, I wish I had a diagram, but I don't. Basically, the way it goes is you need the big picture of the signal initiation is like this. You need to first form receptor aggregates. Then lin can bind to individual receptors as well, but then it doesn't do anything. Lin will cross-phosphorylate. So lin can phosphorylate a receptor that's bound to a receptor that lin is docked onto. So, and it can also phosphorylate a lin that is docked onto a neighboring receptor. So it's cross-phosphorylation. And then, so lin phosphorylates the receptor then becomes more prone to binding. So that means stronger binding between the next lin and the same receptor. They phosphorylate each other mutually. And once there's enough lin and once there's enough phosphorylation in the system, SIC binding can also lead to activation. So lin kinase can do so lin is also represented as a molecular species that can diffuse in 2 dimensions. It has 2 modes of binding to the receptor. One is through something called a unique domain, and one through an SH2 domain. The receptors actually, so each of the items will have an SH2 domain, so the SH2 provides a better binding. However, the SH2 binding only works if the site is phosphorylated. So initially nothing is phosphorylated, nothing is bound. So initially you have just unphosphorylated receptors. They occasionally form a dimer. Then onto this dimer may bind an unphosphorylated lin, which with a low rate may phosphorylate one of the items. Once that item is phosphorylated, it's able to bind the lin more stably. That increases the probability of the lin sitting there and phosphorylating the other item, and also phosphorylating other lins and so on. So that's lin. And finally, there's the second kinase, SIC, which in principle can also bind just on a receptor, just onto a receptor, but it needs to be phosphorylated, and lin can phosphorylate SIC. So lin can phosphorylate SIC as well as a receptor. In both cases, they become more active. SIC Lin can phosphorylate SIC, and SIC can also cross-phosphorylate another SIC, the same way as Lin did. So there's all these phosphorylations and bindings. So all of this comes together in the activation pattern, which, to summarize, so receptors may form dimers and even longer things. Lin, if it's bound to a receptor in an aggregate, then it can phosphorylate the adjacent receptors or lins sitting on adjacent receptors. And I think that, and one receptors can actually bind two lins in the same time because it has the two items. It can so the lin could phosphorylate also the item next to it, but the lin on the item next to it. And finally, SIC that is bound to a receptor in an aggregate can then be phosphorylated by either a lin sitting on a neighboring receptor or by a SIC sitting on a neighboring receptor. So as you can see, so things start from monomer unphosphorylated receptors and free lins and SICs, and then you have to go through a combination of these processes to get to the point that you have a high likelihood of getting SIC activated. So that's kind of, so it's a repeat, it's a variation on the classic pattern of ligand-induced receptor dimerization and cross-activation. It's just there's more players. And that's what makes this a somewhat more interesting problem, because there's all these players and there's all these states. And if you remember, so just focusing on a single receptor. So a single receptor has two sites. Each site has up to 2 phosphorylation states. So those phosphorylation states, so each of them can be in one of 3 states. So there's already 9 states to just the receptor sitting by itself. Then it can bind lin in 2 different ways on 2 different sites, and it can bind SIC at least once. And then the lin and the SIC can either of them be phosphorylated. So there's already a fairly large number of states just to a receptor and things that can bind to it. And now you add to that a receptor can be in any one of these states, and then it can be in a dimer or a trimer with other receptors, which either of them can be in one of these many states. So long story short, I'm sure you've seen many talks about this. There is this combinatorial explosion of quote unquote species. So if I insist on regarding a receptor that's 0 times phosphorylated on the Ig alpha and 1 phosphorylated on the Ig beta, if I insist on considering this as a separate species from a receptor that's just the same except the other guy is phosphorylated, I will have a very large number of species. And because the phosphorylation state does influence the binding properties, but essentially the binding and unbinding is the same thing, I will have an even larger list of copies of the same one reaction. So when a receptor binds to another receptor, it's the receptor binding. It's an on rate, there's an off rate. Okay. It's not supposed to, that on-off rate is not supposed to be influenced by other things. But you have this process of 2 receptors forming a dimer, but because each receptor has 79 different states, it's actually, now you have 79 squared divided by 2, I guess, different realizations of the same exact thing, which is just 2 receptors form a dimer. And the solution to this is sort of a trick to go around this is what they call rule-based. This is a rule-based approach. So as I said, so this is just and then the one thing I should have mentioned about this, in addition to this sequence of events, which is receptor aggregation, Lig binding and activation, and then SIC binding and activation, in addition to the sequence, or this sequence has a lot of little feedbacks. So more lin activation means more activation of receptors. More activation of receptors means stronger binding of Lin, which means more Lin binding, which means more Lin activation, which means more. And there's multiple such feedbacks. So it's really cool to model, but then it's obvious that it's going to be really crazy if you're trying to do ODE based models, because you have this explosion of species. And one of the paradoxes of that is that, you know, you'll see that in our simulation we have maybe a few hundreds of molecules, but even if you can scale it up to 10,000, you easily get to 10,000 species. So actually, you can have, if you have a small enough, or if you have a reasonable size membrane patch, oh, you have 10,000 receptors on the cell. So you can easily have technically more species than you have molecules. So in that sense, paradoxically, having a simulation in which you actually follow the state of every single molecule in your model becomes, well, not so much more expensive than trying to have a comprehensive list of all the species and all the reactions. Okay, and so then, which gets us to our simulation. It's 3:45, so I should probably go faster, but it's almost done. So our simulation or a model is really, it's rule-based, agent-based, and spatial result as advertised. So rule-based is, I just described why rule-based. So basically, you have these many processes which all occur in many similar state, with similar substrate, similar between similar molecules, which are just differ by 1, 2, 3, or more other features that are not crucially influencing they're not influencing the existence of the reaction. And so then you have duplicates of reactions. And so the group of all these duplicates is this meta reaction, which is called a rule. So rule is that, you know, two receptors that are not bound together, and they have, each of them has at least one free receptor binding domain, can form a dimer. That's a rule. And so instead of individual reactions, we have rules. And the model is agent-based in the sense that we have these three base species, the receptor monomers, Lick monomers, Sick monomers, Lin monomers, Sick monomers. Of course, you form aggregates which have more than one of these base items together. But the trick is that we have this fixed list of receptors, Lin and Sick molecules. Each of them has an identity. So we actually, and what we do is we keep track not only of the position, but also, or the phosphorylation state, but also of the binding state of each of them. So the variable types that we use to describe receptors, Lin or Sick, also have slots for the identifier of the Lin, Sick, or other receptor that they're bound to. So this way, memory-wise, we don't, it's fairly economical. So you don't have to, all you have to do is, you just have to keep track of essentially the ID. So you have a list of receptors with IDs, list of Lins with IDs, list of Sicks with IDs. And then the receptor variable has two slots for the ID of the receptor that's bound to it, and so on. And so that's the way we keep track of the binding and phosphorylation states. And we also keep track of the physical position of things. There's a trick to that. I'll get to that. But that's it. So it's rule-based, agent-based, and I'll explain how the spatial stuff goes. Now so this is a detail. I'll skip over this. That's why it's called a detail about the rule-based stuff. It's an example. So the spatial motion. So why do we need the spatial thing in the first place? Well, because, I mean, dimerization. So the key reaction at the end of the day is dimerization, which is triggered by the fact that two things collide. So when you have 100 things in a box, they're not equally probable to all dimerize with each other. It depends on where they are, right? That's one thing. And the other thing is that, well, we are actually trying to leverage spatial data, right? So we really are emulating the thing that you can see in the single particle tracking. So long story short, we actually have a spatial representation. So we represent this as a reaction diffusion system. So the reaction part is agent-based in the sense as I described, but the diffusion part is just like in Smolden. I'm sure someone here knows about Smolden. It's a big enterprise. I mentioned Andrews and Bray from 2004. That was the original, but it became this big, so it's still around. They said, so again, this came out last year, but it was started fairly long time before that. And what we're trying to do is essentially something that does both what Smolden does and what these rule-based simulators do. And so it's kind of a one-off dedicated thing. So the key element is that so in addition to regular diffusion simulation, which basically means Brownian motion, which basically means that every particle at the appropriate time is given a randomly selected jump distance and direction, and it jumps there, and then you check for collisions. In addition to that, one salient element of the Smolden approach is that reaction on rates are characterized by something called a binding radius. So a dimerization reaction or an interaction-based reaction is triggered by collision. So it's triggered by the two participants being, well, colliding. But remember, what we have is point particles. We represent them as points. So then depending on the rest of the details of the simulation, on your time step or your diffusion constants and all those other things, the probability, so the on rate for a binary reaction that's in the literature translates into a value for the binding radius. So the binding radius is basically the distance. So if A might dimerize with B, then during the simulation, you move A, you move B. At one point, you check for collision. So you check whether there's a B at distance D from the A. And the rule in this type of simulation is that, well, if the distance is less than the binding radius for this reaction, then A will dimerize with B. But that means that the binding, so if you look in the paper, we list the binding radii, which we derive based on rates from the literature. Okay. And there's another more technical thing, which obviously we don't have time to go into, which is the unbinding radius, which is a whole different thing. Basically, it's when that dimer breaks up, where do you put the products? If you put the products at the binding distance, then at the next iteration, they will have to dimerize back again, which is stupid. So you want to put them further up. It's a different thing. All right. And so that was the other technical thing. And finally, the microdomains. So the microdomains, as I mentioned, they're basically derived, they can be derived from just looking at the trajectories. And we actually derived some microdomains shapes, and we put them into our simulation. And so finally, on to the results. So this is a schematic of the simulation space. It's 1.5 microns across squared, and there's a thin layer. So the thin layer is only there because of SIG. So we also have the trick there, but basically we have a fairly thin layer, and then we have a well-mixed box. We say that the layer is in contact with a well-mixed box, and so there's a probability of things coming out from the box randomly showing up in the layer, in the far end of the layer, and there's a probability of the particles in the layer diffusing into the box and then losing their position. Okay. And otherwise, it's kind of this. So we start with free receptors in the patch, free receptors and free lane in the patch, and then they will collide and form dimers. The dimers may break up. They may collide and bind the lin. The lin may drop off, and once in a while, there's a probability per time for all these phosphorylations and cross-phosphorylations occurring. so the unary reactions that don't rely on collisions are implemented are characterized by a rate of occurring. And so they can be simulated with a Gillespie type simulator, except that the diffusion needs to be fixed time step. So what we do is we actually convert the rate into a probability over the time step, and there's a way of how you loop through all the particles. So basically there's a small time step update in which you move only one particle, you check for its collisions. If there's no collision, if there's a collision, you implement whatever. That collision says it's a dimerization. If there's no collision, then you compute the total probability of any unary reaction happening, and then you pick the unary reaction like you pick them in Gillespie, and you implement that and you move on. So that's kind of it. And so as I mentioned, so all the parameters are from the literature other than the ones that we could get out of SPT, which is diffusion rates and off rates. And even on rates, I think. Diffusion rates, on rates, and off rates. I think so, yes. And so here are some results. So we basically performed four types of runs. So we have two cell lines, and we ran them with domains and without domains. So one of the big differences, both are malignant, but one of them has a much higher dimer lifetime, because that's the norm6. And so this is the distribution of aggregate sizes for the same given number of receptors. And as you can see this, so if you have no domain and you have the less persistent dimer cell line, then you get mostly monomers, a few dimers, and nothing else. But if you have domains and or you use the other cell line with a longer dimer lifetime, then you end up having even more higher than 4 aggregates. This is kind of an example of what the time courses look like. So it's basically where this counts the receptor activation over time. Again, you can see that it's on the left you have the domains on the right no domains, and the short lifetime is short dimerization, is dimer lifetime is at top, long dimerization is at the bottom. So this one, as you can see, has more phosphorylation. No, sorry, this one on the left has more phosphorylation events and longer than this one, as you can see. Similar differences persist. So one thing to be said here is that in terms of phosphorylation, this is receptor phosphorylation. The dimer off rate has a stronger effect than the presence of the domains. The same thing translates into Lin binding and activation, and finally SIC. With SIC, so that's the final step, that's the signal you're looking for. So there's barely anything happening in the short lifetime version. There is some signal in the long lifetime version, but that kind of makes our point that, well, yes, so just the assumption that everything with pre-BCR signaling goes the same as with BCR signaling, except for the very short lifetime of dimers. That assumption, and putting it all together best as we could given the resources, does indicate that yes, you can have that does actually explain the existence of tonic signal. Now, why these cells are around and they didn't die, that's a whole different story. That's not part of this. So thank you for your patience. Oh, that's the SIC signal. So as you can see, the norm6, so the effect of the long lifetime is stronger than the effect of domains, but domains also make a difference. And that's that. What I just said. And so support, as I said, this is was primarily in the UNM STMC. So I'm at West Virginia University, but I've been working with them for a long time. Had this grant with Jeremy Edwards is like main kind of usual suspect, and thank you for your patience.
Okay, thank you very much. Time for a question or two.
Let me just unshare this.
Anybody?
Yeah.
If anybody has trouble unmuting, let me know. So I think, well, we're, sorry, Jim, was that you? Did you have a question? I thought I saw a hand raised.
I don't see.
Sometimes people have to leave at
Yeah.
4. So I think it's very interesting to hear this conversation about the difficulty of trying to do sort of classic reaction kinetic type modeling, the need for rule-based approaches. And when you implemented this, maybe you could tell us what were the biggest challenges you faced in actually implementing this very complex model computationally.
Well, I mean, one thing is that it's kind of slow. That's one thing. Other than that, I think it's I'm the one who's I'm not I'm no I'm not against it at all, but I'm not I'm not against things like um, making software for other people to use. But I think the complexity of it and the fact so my experience is that by the time I would figure out how to use Bionet Gen in the latest version and the way it's combined with Smallin, which supposedly it is already, it's easier for me to just write my own simulation. But on the other hand, obviously that's inefficient. So that is very frustrating that there should be some kind of intermediate way of doing this. So there should be some sort of intermediate way of well, so you want to generate the so when you want to generate the so we all of this was done by hand, as in defining the object that so each species will have to have a different object, right? So it will have to have phosphorylation states. It will have to have it will have to have a pointer for each of the things it can bind to. You will have to have these cross checks. So basically, we have this double accounting. I don't know what if that's the proper English term for it. So each receptor knows the name and address of every lin that it binds to it. Each lin knows the name and address of each the receptor it's bound to. So then you basically every time you make a link, you have to note it on both variables. So I think what's most frustrating is constructing the variables. And then the other thing is, I think that the way Smallin, or I shouldn't use the term Smallin, but the way this Smallin style algorithm works, and you're physicists, right? It's like renormalization of it, because so the problem there is that if we do fixed time step, right? In a finite time with true Brownian motion, there's a non-zero probability that a collision occurs over any finite time, no matter how small. So you're always glazing over possible events. Now, the reason it's okay to glaze over possible events is the paradoxical fact that, well, actually, it's not really Brownian motion. Brownian motion is the approximation of something that's more like collisions and stuff. But that means that the scheme where you have a binding radius and an unbinding radius you know, on the one hand, you can argue that it's mathematically incorrect because you're glazing over collisions and all that. But that has just about as much reality content as the model that so Brownian motion is an idealized model that approximates some real situation. Now you do a simulation that approximates the idealization. And so I had this, if I would rant about things, I would say that, well, if you're going to be in this, if you're going to do this, then you should, then it's kind of useful to point out that the simulation itself might be just enough to define the reaction, it's the physical process itself, because you don't know all the details. You don't have all the details. Anyway, that's what I'm saying. What's frustrating is that most people don't grasp, don't appreciate things like this.
Well, when you have these stochastic processes, and you have some things that are synchronous and some things that are async, it's hard. I mean, if you have to have if you have to do something, have an event every time a single thing happens, and the probability of multiple events within any time window is non-zero, then, as you say, it's a hard problem. And so it's impressive that you're able to get it to work. It's not, it's really a complex model. It's a very complex model. Lorenzo, go ahead.
Hello. Thank you for your talk. It was really nice. I really liked all the details, etc. My question was actually, say that if we can imagine that in your system you can identify some measurable that goes in and some measurable that goes out, right? Can we eventually, after your results, obviously, all your experimentation and tests, could you eventually find the boiling down the system to some functional form? You mentioned that ODEs might not be adequate because you have to do too many things, right? If you have to do ODE for every single detail. But after your experience with this model, did you find, or did you have an intuition that perhaps the whole system that you're describing could be captured by some approximate functional form? So to simplify everything in one, not say a rule will be a little bit perhaps too harsh, but perhaps a functional form that is slightly more complex than linear, or just that approximate, or perhaps some parameter like the ski or the lean thing that might give different outcomes dependent on those parameters, rather than a lot of parameters that obviously you have described to detail everything.
So I can only say that I think I'm exactly where you are in that I realize that, so I think that's the not a paradox, that's like one of the issue with all these over detailed simulations. So it's detailed, but it's obvious that there's so many things that can be corrected or can be questioned about it. And so ultimately you can make it work, and the best thing you can do, you can make it work and kind of match everything that's measured, right? And then you can, well, so there I would suggest that we being people who do simulations like this, I guess, we might, or people who, or no, and also people who are looking at computational ways to gain insight into this type of systems. So we might just say that, look, the ODEs are a model, the simulation is a model. If the simulation works and it's calibrated, that's as good a model as you can get, and not think about it. And I think what you're suggesting, Lorenzo, is the next step is to say, okay, well, so now can I take the model and get an approximation of that, which is not as expensive as rerunning the model, and then I can encapsulate that into signaling models or other things. And I'm saying I'm there. I recognize the need or the utility of that, but I think is just like this. It's a lot of work, and you need some sort of a framework and well thought out and carefully thought out goals to not waste your time for another for 2-3 years on something that everybody else will say, yeah, what? Tell me something I didn't know. That's why.
One other sense is difficult when we have the other domain. I come from the biology domain, so it's I'm a cell biologist, etc. And I have an idea of what are the kind of data that you get from that area. You can get sometimes serious in the best of luck, but very often from clinical samples you get just, phenotype, things that are kind of. So it becomes very difficult to drill down this data to the resolution that you are describing, which is essentially down to the Brownian motion of molecules that interact with each other. So then it becomes extremely difficult to map to some outcome. I'm not saying that, you can create, well, we hope we can one day create a system so complex to model a disease. But I think it's important, well, both to do the hardcore job that you illustrated, it was really nice, and trying to synthesize probably so that we can map better to the data that we have available, because these are very often a little bit more coarse.
But I mean, okay, that's I'm not so if you think of how this, so I've been in this since for so I think for about 20 years now, I think. Because I'm a nuclear physicist originally. So what was at the time, so late 1990s, early 2000, what drove it was that, well, you had the sequencing, so the whole genome sequences coming out. So, and then you had these physical methods that were able to produce more data than before. So you had more direct information on the processes. And of course you had more, wider spread of computing. And that's kind of because I think the difference between biological systems and physics, what physics studies, is that physics really studies simple things, very simple things, which can be separated, and biology doesn't separate well. And so then the possibility there. So my point is that any progress in quantitative biology or in this kind of first principles, so going back to first principles rather than let's just look at probabilities of what is it, any progress in this direction was always made possible by computing. So by moving what a person would do, like emulating that and getting a computer to do that 1,000 times faster. And so maybe that's the way out here as well. So so there are highly detailed molecular calculations of this and that, and they can be done, but it's just unrealistic to expect to do them for every single thing. But maybe I think that's something people do. So they try to kind of approximate, so find some ways of approximating the outcome of highly detailed calculations. So replace that with some sort of a lookup table or just an approximation, and then use that cheap approximation in many different instances simultaneously, and then reason about them. And by the way, machine learning does that. So that's what it does. Now, how to put these together, the problem is, I think, that you always have, so there's a true low-hanging fruit, and then there's a lot of false low-hanging fruit. There's a lot of false things that seem very easy, and let's just put it in, and you seem to get some result. But I think ultimately there should be some way of taking all the detailed systems biology and computational systems biology, pre-machine learning and AI, and somehow leverage that with AI, not replace it. I don't know how that's.
So other questions. I guess to follow on to that, did I understand right, the two cell types that you are modeling, The only difference in input parameter your model is the lifetime of the dimers. Is that the only difference that
Yes,
you put?
and maybe the diffusion rate, but I think it's the lifetime of the dimers, yes.
So in principle, you're going to have some dose response, which is if I input to this complex model the diffusion rate of the monomers, for example, or the dimers, and the lifetime of the dimers, then it's going to predict the sick activation level.
Yes.
And so in principle, Lorenzo's point that here you have a homogenization that you want. You fundamentally want something where you have a table which says, here are these two measurable parameters, experimental parameters. I want to predict the level of activation. Now I'm doing it through quasi first principles molecular dynamics methods. But in principle, the outcome, if you could do lots and lots of simulations with lots and lots of different lifetimes, would be essentially a transfer function that says, given the lifetime, give me the sick level.
Right. Yep.
If I could run it enough times over a long enough period under enough conditions. And then I might say, well, I also want to know what the domain size, the microdomain size is, for example. That might be an input parameter. But you could imagine that here, there are not that many things that are going to be different from cell to cell. They might be hard to measure experimentally, although in principle, Lorenzo, these are cells that you could take from a patient, put into, it would be an expensive measurement, but in principle, you could do this measurement. These are cell lines from patients. So in principle, it could be a patient-specific measurement that if you wanted to. And so I think actually this is a nice example because the homogenization, the inputs and outputs are pretty clear. And so actually I think that this may be a case where the problem of having to model everything is not as bad as it could be in some other cases.
But they only have these two cell lines.
Well, you'd have to, but in principle, you could pull patient-derived cells, make the same diffusion measurement, and then put that into your simulation and say, well, our calculated
right. So and that would be I mean that would kind of be a next step here. Yes, I absolutely agree. And I think that's that's the one little twist to that is that I think it's that they were looking for, so what seemed more important was to find the mutation that resulted in that difference of lifetimes, which is kind of so physicists would say exactly, I mean that's the physicist mindset. Okay, fine, so let's make this into a function and let's try it. But it's, as you know, it's not trivial to get the cell lines, and even so, it's not trivial to get the cell lines because they would actually have to come from patients, and then it's not trivial to repeat this experiment 100 times.
But your simulation can predict experimental outcomes.
Right,
you can say if you pulled a cell from a patient and you make the measurement, then the expected sick level of activation would be this.
Yeah, that is.
So you now actually can make a prediction before they do the experiment.
Or it would be a better, or at the very least, it would be a validation or refinement to the model because it might not end up working that way.
And of course, then if you have something where the treatment options depend on the sick activation level because you pick a different chemotherapeutic, then it becomes, I mean, you can see how this fits very nicely into a genuinely patient-specific workflow.
No, I think that was the
goal. Yeah.
The goal, and I think it's not that kind of so this was so the last, I think the last Bridget is the last, so Bridget Wilson retired, I think. What's the I think I have the, I'm not sure which one. So Jeremy is he's around, but he's more into sequencing now, and Bridget retired, and Frank and Ramika both went to industry. that's what this is. But the groups are still there, and I mean, that would be a, I don't know, do you know of that? So that was certainly the idea, but I think it's that this should be countered by, so if you're looking at a proposal, then okay, so here's Adam proposing what you just said. I'm gonna run this thing. I need some computing power to refine this and get constructed distribution. But once I have the distribution, then everybody can just come and run their cells, and so that should be countered with what's the alternative, and I think that's the alternative. What is the alternative?
It may be easier to measure the SIC activation level directly than it would be to measure the diffusion constant.
Exactly, right. And that I think is the problem. So you would have to essentially pair up with someone, and you didn't ask me what is the SIG activation level, and I think the answer is that they didn't measure it. But basically you would have to pair up with someone who is willing and has the ability to do the SIG activation level and simultaneously do this, but then it would be really all about measuring the SIG activation level, which is okay, but right, and then it would be, so then you measure the SIG act. Well, I think that's a discussion about what is the utility of, because then this makes this an example of why you would still want to, support people making a big computational model like this, because ultimately you will never have more than just until this becomes a thing, you'll never have more than just a handful of cell lines.
Right. Well, clearly you want your inputs to be things that are easier to measure than the things they're doing now for the targeting. I'm not saying that this specific example is going to work, but I think at least as a proof of principle about thinking about these problems, and Lorenzo's comment about the fact that if you undid enough of these simulations and were able to validate them, that at least conceivably, if one came up with a way that was easy to measure that diffusion constant, then you could imagine this being a very clinically meaningful measure, calculation. Even if you weren't doing the microcalculation every time, the fact that you could do it in the background is already very interesting. So I think this is really nice. I think we've overrun our time a little bit.
Yeah, I'm sorry.
No, it's fine. It's a complicated model. It's unfamiliar to people, so taking the time to explain it clearly is not a minus at all. I just want, is it Itamar, Sharon, Lorenzo, do you have a last question for our speaker today? No.
Not me. Thanks for a good talk.
Okay. Well, I think it was really great. We don't hear that many sort of MB scale talks, and the combination of rule-based modeling with explicit diffusion and reaction, to me, it's very exciting. I really appreciated it. And I appreciated the time you took to lay out what I would call the conceptual model very clearly.
Wow, thank you.
And so I do appreciate you coming to speak to us today, and I hope you find ways of bringing this forward. And you're welcome to come and listen to other talks in our series too, if you like.
I'll check in. Yeah.
Okay.
Well,
Well, I
thank you
thank people for sticking around, and I'll see people in 2 weeks. Thanks so much, everyone.
Thank you very much. Have a good weekend. Thanks for having me.